For much of the last century, the story of human evolution has been told with a sense of quiet resolution. Our species emerged in Africa, developed the traits we recognize as fully human, and then spread across the world. Along the way, we encountered other human forms, Neanderthals, in Europe and Western Asia, Denisovans, farther east, who lived for tens of thousands of years before disappearing. In this familiar account, those groups are often described as having revived for a time, adapted to harsh environments, and then vanished, leaving behind little more than fossils and stone tools. This understanding shaped textbooks, documentaries, and museum displays. Neanderthals were portrayed as a separate branch that went extinct around 40,000 years ago, replaced by modern humans who were somehow more adaptable or more efficient. The boundary between them and us seemed clear. Their story ended, ours continued. Yet even as this picture settled into place, there were details that never fully fit. Archaeological layers showed long periods of overlap. Some tools looked unexpectedly similar across population. Certain fossils resisted easy classification, a priori, knife fully. Nasistence. What no one could see at the time was the deeper record carried inside bone and tooth. Locked away at a scale far smaller than any but for decades, that record was inaccessible. DNA, fragile and easily destroyed, was assumed to be beyond recovery once thousands of years had passed. Human prehistory remained a story reconstructed from shapes, measurements, and careful inference. That assumption changed quietly, without fanfare. As technique improved and fragments of genetic material were finally recovered from ancient remains, a new source of evidence entered the conversation. It did not replayer that could not be seen before. Ancient DNA began to reveal patterns of ancestry, contact, and survival. That no fossil alone could show what emerged from this genetic record was not a simple confirmation of the old story. It was something more subtle and more entirely disappear. In a biological sense, parts of them endured. Their legacy did not end in the distant past. It continued quietly within the gums of people living today. This is the point where the familiar story begins to shift, guided not by speculation, but by evidence written deep within our own cells. Before genetic evidence entered the picture, knowledge of Neanderthals was built almost entirely from what could be seen and touched: fossil bones, stone tools, and traces of ancient campsites. From the foundation of interpretation, from these remains, research and reconstructed bodies, behaviors, and time mimes. By the mid-20th century, a general consensus had formed. Neanderthals were recognized as a distinct human population that evolved in Europe and Western Asia after an earlier migration out of Africa. They were adapted to cold climates, with stocky bodies, broad chests, and powerful limbs. Their skulls were long and low with pronounced brow ridges. These features were understood as specializations for Ice Age environments rather than signs of primitiveness. Archaeology showed that Neanderthals were capable toolmakers. They produced sophisticated stone tool traditions, hunted large animals, core attempts ranging from Mediterranean. Over time, the older image of Neanderthals as slow, brutish creatures was replaced by a more nuanced view of a capable and resilient human form. Despite this growing respect for their abilities, the dominant narrative still placed Neanderthals on a separate evolutionary track. The prevailing model held that modern humans evolved exclusively in Africa, and later expanded outward, replacing local archaic populations with little or no genetic mixing. This framework became known as the recent African origin model. In its strictest form, it allowed for cultural contact, but rejected biology. Under this view, Neanderthals and modern humans may have encountered each other, but they were not thought to have merged. Any similarities in tools or behavior were explained as imitation or coincidence. When Neanderthals disappeared from the archaeological record around 40,000 years ago, climate, instability, competition for resources, or demographic vulnerability, but the outcome was the same. Neanderthals were gone. This separation was reinforced by anatomical comparisons. Neanderthal fossils showed consistent physical differences from early modern humans found in Africa, and later in Europe, if Neanderthals had interbred extensively with incoming populations. Many researchers reasoned the fossil record should show a clear blending of traits. Because such blending was difficult to demonstrate conclusively, the assumption of limited or non-existent interbreeding persisted. There were dissenting voices, but they remained on the margins. Some anthropologists pointed to fossils with mixed features or to long periods of overlap between populations. Others suggested that complete replacement seemed unlikely, given the small sizes and mobility of ancient groups. Still, without a way to test genetic continuity directly, fossils are rare, unevenly preserved, and often fragment entire populations can be represented by a handful of individuals. Stone tools, while informative, reveal little about biological relations. From this incomplete record, researchers were asked to reconstruct population histories, spanning hundreds. Caution often favored simplicity. It was easier to describe Neanderthals as a branch that ended than as a population that partially merged. Extinction, as a concept, fit neatly with the evidence available at the time. The absence of visible continuity was interpreted as proof of disappearance. By the late 20th century, this understanding had become deeply embedded. Neanderthals were acknowledged as intelligent and human-like, but still separate. Their story was presented as a parallel experiment in human evolution that ultimately failed. Modern humans, in contrast, were portrayed as the sole survivors, carrying forward an unbroken lineage from Africa to the present. It was a framework built carefully, logically, and with the best evidence available. But it rested on an assumption that would soon be tested in a way no fossil ever could. The idea that genetic material could survive for tens of thousands of years was once considered unrealistic. Moisture, bacteria, and time itself break it down into tiny fragments. For most of the 20th century, researchers assumed that whatever genetic information ancient humans once carried was irretrievably lost. But not ancestry in a molecular sense. That assumption began to change in the late 20th century, as laboratory techniques in the late. Scientists learned how to work with extremely small and damaged pieces of DNA. They developed clean room environments to prevent contamination, along with chemical methods that could isolate and amplify genetic fragments. What emerged was not intact fragments, but scattered sequences, short stretches of genetic code. The earliest successes came from relatively recent remains preserved in cold or dry environments. But even these were difficult. Ancient DNA is often outnumbered by modern contaminants, including bacteria and traces of DNA left behind by archaeologists themselves. Distinguishing authentic ancient sequences from noise required careful controls. A major turning point came with the focus on dense skeletal elements, especially the inner ear bone known as the petrous portion of the temporal bone. This structure protects DNA better than most other parts of the skeleton. Teeth, particularly their roots, also proved to be valuable sources. With these materials, researchers began recovering genetic data from individuals who had lived tens of thousands of years ago. At the same time, high-throughput CNN transformed what was possible. Instead of targeting a few specific genetic markers, scientists could now read millions of fragments. Computational methods were developed to assemble these fragments, identify characteristic patterns of ancient DNA damage, and compare them to large database of modern human germs. The first successful extraction of Neanderthal DNA came from remains found in Europe, including sites in Croatia and the Altai Mountains of Siberia. These individuals lived roughly 50,000 years ago, near the end of the Neanderthal timeline, when their genetic sequences were compared to those of living humans. The results were unexpected. Rather than forming a completely separate branch, Neanderthal DNA showed clear points of overlap with the genomes of modern people. They followed consistent patterns that suggested shared ancestry rather than coincidence or contamination. The signal was subtle, but it was unmistakable. To test this, researchers compared genomes from different modern populations. Individuals with ancestry in Europe, East Asia, and the Americas consistently carried small percentages. In contrast, populations with deep roots in sub-Saharan Africa showed little to no such signal. This geographic pattern was difficult to explain without invoking interbreeding. The timing also mattered. Genetic modeling suggested that this exchange occurred shortly after modern humans left Africa. At that point, expanding human groups would have encountered Neanderthals in the Middle East and surrounding regions. The genetic evidence indicated not just contact, but successful reproduction. Importantly, this was not inferred from a single independently or site. Neanderthal germs recovered independently and analyzed by different teams. The findings were done back across laboratories and supported by statistical tests designed to rule out alternative explanations. Ancient DNA also allowed researchers to estimate population size, and Neanderthal germs showed signs of long-term small population size and repeated bottlenecks. Their genetic diversity was lower than that of early modern humans, suggesting vulnerability to environmental change and demographic instability. At the same time, the segments of Neanderthal DNA found in modern people were fragments, indicating that many generations had passed since the original interbreeding events. If Neanderthals had contributed genes recently, the inherited segments would be long and continuous. Instead, they were short and scattered, consistent with ancient mixing followed by thousands of years of recombination. The genome itself preserved a timeline, as more ancient Germans were sequenced, including those of early modern humans from Eurasia. The picture became clearer. Some early individuals carried higher proportions of Neanderthal ancestry than people today, suggesting that Neanderthal genetic contribution was once more substantial. Over time, natural selection and demographic process reduced its overall presence. Each genome added detail, confirming that Neanderthals were not completely replaced. They were absorbed, in small but measurable ways, into the populations that followed. This genetic breakthrough did not invalidate decades of archaeological. Instead, it reframed it. The fossils and tools still mattered, but they now existed at a locator level. Together, they pointed toward a more complex human past, where extinction and survival were not always distinct outcomes, and where disappearance did not necessarily mean genetic erasure. As the genetic data accumulated, patterns began to emerge that challenged long-standing assumptions about Neanderthal, extinction, and modern human origin. The most striking revelation was not simply that interbreeding occurred, but that Neanderthal genetic material persists in a structured and uneven way across modern populations. This was not a trace left by chance. It reflected specific encounters, migrations, and demographic events that unfolded over thousands of years. On average, people living today outside Africa carry about 1 to 2% Neanderthal ancestry. That number may appear small, but when spread across billions of individuals, it represents a substantial genetic legacy. Importantly, this ancestry is not uniform. East Asian populations tend to carry slightly higher proportions than Europeans, while indigenous peoples of the Americas show similar levels shaped by their Eurasian origins. These differences suggest that interbreeding did not happen just once. It likely occurred multiple times into regions already occupied by Neanderthals. Genetic comparisons revealed that Neanderthals themselves were not a single homogeneous population. Groups living in Western Europe, the Near East, and Siberia showed genetic differences, reflecting long periods of separation. When modern humans encountered these groups, they did not meet a single Neanderthal population, but several related ones. The DNA also revealed when and where these interactions most likely occurred. The strongest signals point to the Near East as an early contact zone, where modern humans exiting Africa encountered Neanderthals, who had lived there for hundreds of thousands of years. From this region, human populations dispersed both westward into Europe and eastward into Asia, carrying Neanderthal genetic material with them. Later encounters in Europe and possibly Central Asia added further complexity. One of the most important insights came from examining which Neanderthal DNA is not evenly distributed across the modern human genome. Certain regions are almost completely devoid of it, while others show a higher than expected concentration. This pattern indicates the action of natural selection. Some Neanderthal genes were likely disadvantageous in the long term, These tended to be associated with male fertility and early development. This process helps explain why the overall proportion of Neanderthal ancestry declined. Yet other genes persisted, and some may have provided adaptive benefits. Several surviving Neanderthal genetic variants are linked to immune function. These genes may have helped early modern humans respond to pathogens encountered outside Africa. In unfamiliar environments, where local diseases posed serious risks, inherited immune responses, shaped by long-term Neanderthal adaptation, could have offered a survival advantage. Similarly, genes related to skin fesolar climates and lower levels of sunlight. The DNA also revealed the demographic vulnerability of Neanderthals themselves. Their gums show extended regions of low diversity, consistent with small isolated populations. Over thousands of years, repeated botulinus reduced their genetic resilience. When modern humans arrived in larger numbers, even limited interbreeding could have had an outsized effect on Neanderthal population structure. In this context, Neanderthal extinction appears less like a sudden disappearance and more like a gradual absorption combined with population decline. Ancient DNA from early modern humans added further detail. Some indicate higher levels of Neanderthal ancestry than any population today. In a few cases, genetic evidence suggests that a Neanderthal ancestor may have lived just a handful of generations back. These individuals did not belong to a separate hybrid population. They were fully integrated into modern human groups. Over time, as populations expanded and mixed Neanderthaler segments, each generation reshuffled the genome, fragmenting the inherited sequences. What remains today is a mosaic, thousands of small Neanderthal-derived fragments spread across the genome, different in each individual, but collectively preserving a record of ancient Neanderthals. Crucially, the DNA evidence showed that Neanderthals did not survive as a distinct population. There is no indication of a hidden, isolated Neanderthal group lingering in remote regions. Instead, their survival was genetic rather than cultural or demographic. They persist not as a separate people but as contributors. This realization forced a redefinition of extinction itself. In evolution forced a redefinition itself. The boundary between replacement and continuity became blurred. Neanderthals were not simply replaced by modern humans. They became part of them in small but enduring ways. Taken together, these findings transformed the narrative. Human evolution was an and-rated process, shaped by movement, contact, and exchange. The genetic record did not depict a single victorious lineage sweeping aside all others. It showed a history of interaction, where survival could take unexpected forms, and where disappearance did not always mean the end. The realization that Neanderthals survive within modern populations reshaped more than a single chapter of prehistory. It altered how scientists understand what it means to be human. The genetic evidence made clear that modern humanity did not emerge in isolation, sealed off from other human forms. Instead, our species was shaped through contact, interaction, and limited but meaningful exchange with closely related. Neanderthals were not an entirely separate experiment that failed while modern humans succeeded. They were part of a broader human landscape in which multiple populations adapted to different environments and occasionally met. When they did, those encounters were not always hostile or fleeting. The persistence of Neanderthal DNA also reframed the concept of ancestry. For people outside Africa, ancestry is not a single unbroken line leading back only to early modern humans. It is layered. Within each genome lies a record of ancient journeys and encounters, compressed into fragments too small to notice without modern technology. These fragments do not define identity, but they remind us that human history is collective rather than isolated. From a biological perspective, the findings helped explain subtle variations among populations, difference in immune response, skin filaments, and metabolic. They were not dominant influences, but they were part of the genetic stochastic early humans carried as they adapted to new regions. At the same time, the limits of Neanderthal genetic survival are just as important as its presence. The fact that most Neanderthal DNA was graphersons between populations. Natural selection favored some inherited traits and rejected others. The result was not a blending of two equal populations, but a selective integration shaped by long-term survival pressures. These discoveries also encouraged greater caution in how extinction is discussed. The disappearance of Neanderthals as a recognizable population. But extinction did not mean total genetic loss. The human past contains other examples of this pattern where populations vanish demographically, yet leave traces within those who follow. Ancient DNA made this process visible. First time, perhaps most importantly, the Fungus reinforced forced distance. They were close relatives, separated by geography and time rather than by a fundamental biological divide. The genetic distance between Neanderthals and modern humans was far smaller than once assumed, and small enough to allow to diminish the uniqueness of modern humans, nor does it elevate Neanderthals beyond the evidence. It places both within a single complex story of human evolution shaped by migration, isolation, and reunion. In that story, survival is not always visible in bones or tools. Sometimes it endures quietly, carried forward in fragments. Generation after generation, the recovery of ancient DNA changed the way the human past can be studied. Not by overturning everything that came before, but by adding a deeper layer of evidence. Fossils and artifacts still define the outlines of Neanderthal life, their bodies, their tools, and the environments they inhabited. What genetic evidence provided? It revealed how populations that seemed to vanish could persist in ways that were previously invisible. Neanderthals did not survive as a people, and they did not shape modern humanity in sweeping or deterministic ways. Their genetic contribution is limited, fragmented, and uneven. Yet it is real. It demonstrates that human evolution was not a sequence of clean replacements, but a process shaped by repeated encounters and shared history. The line between extinction and survival is thinner than once believed. Ancient DNA also demonstrated the power of molecular evidence to test long-standing assumptions. Ideas that had remained unchallenged for decades could now be evaluated directly against the genetic record. In doing so, researchers gained a more accurate picture of population movement, interaction, and vulnerability. The story that emerged was quieter and more complex than earlier models, but also more consistent with the evidence. Each new genome adds resolution, filling gaps that fossils alone cannot address. Ancient DNA does not offer a final answer to human origins, but it provides a framework grounded in measurable inheritance. It reminds us that the past is not entirely gone. Parts of it remain present, carried forward. The survival of Neanderthal DNA in modern populations is not a dramatic revelation. It is a careful one. It speaks to continuity across deep time, to the closeness of human populations once thought separate, and to the value of evidence that can endure long after cultures and landscapes have changed.